Hello, my name is Beate Samperson. I'm super excited to be here today at this webinar. And I'm actually joined today by Ellen Verhoef. We both and we are work at the Max Planck Institute for Psycholinguistics, but we do hear ourselves with a 20-second delay. Put the sound off to the sound. Just give us a second as we need to switch off our. So I hope this works better and you can hear us much better because this is important. Yeah, we are very excited to be here today and talk and discuss a little bit about our research on deciphering the genetic links between early language abilities and ADHD. So starting again, my name is Beate Samperson. I work at the Max Planck Institute for Psycholinguistics in Nijmegen, and I lead there the Population Genetics of Human Communication Research Group. Again, an introduction. I'm joined today by Ellen Verhoef. Ellen is a postdoctoral researcher in my group, and we have done a lot of research on early language and want to show you today what we found. Good. I hope you can all see now the first title slide of our talk, and we will guide you through our research step by step and be taking it in turns to explain you what we think is behind the links between early language and ADHD. So first again, we show you a little bit of where we are situated in Europe. And you can see us there really in the Netherlands. And this is a short overview of our wonderful institute, the Max Planck Institute for Psycholinguistics. This is our group. Our research focus has quite a spectrum. Using genomic tools, we disentangle the developmental processes underlying early language development. We investigate social behavior in different contexts. We study overlap of language and social traits of autism and ADHD. And we also sometimes develop structural models to better understand these developmental changes. So today, we really focus on the question how we can use genomics to understand language abilities and the links with ADHD. Starting with the last bit, language abilities and ADHD. It is quite known that children with ADHD experience challenges to acquire language abilities, such as to produce and understand language. They also experience challenges with literacy abilities, such as reading and writing. And research has shown that up to 40 children diagnosed with ADHD have also developmental dyslexia. So there is a bit of a link between ADHD and language and literacy, most often conducted by research covering language and literacy in mid-childhood and adolescence. So what does genomics tell us? And I now hand over to Ellen to tell you a bit more.
So thanks very much. And I think before we really delve into the details about what's linking language and literacy to ADHD, I wanted to tell you a bit more about genomics. But we also just saw a viewer comment coming in about translators having ADHD, and that Ruth actually knows a lot of other translators with ADHD, and she would love to learn about this topic or be a guinea pig. I actually don't know much about this, also not about like the prevalence, but I think it could be a very interesting idea to see whether there's actually this increased prevalence and why this could be. So thanks a lot for that comment. So then I want to go on and tell you a bit more about genomics and some of the concepts that we come across in our research. So when we talk about genomics, we actually talk about the information that is stored in our DNA, which we carry in the form of chromosomes in all of our body cells. And we have actually 23 pairs of chromosomes, of which we inherit one from our mother and another copy from our father. And when we unravel a bit the structure of a chromosome, we see here the double helix structure emerging, which is quite characteristic for DNA. And when we zoom in further, we see the actual building blocks of our DNA emerging. And these are 4 different chemicals, also called nucleotides or bases, that are represented by 4 different letters, A, C, T, and G. And every single cell in our body contains a complete copy of our genome, which in fact is approximately 3 billion DNA base pairs. And when we tell you about studying genomics, we mean that we study all of this genetic information within a person. Then, now you know this, you may wonder, so how does this relate to differences between individuals? So while we carry actually the same genetic code at the majority of places in our genome, there are these instances where we actually do differ from one another. And when this involves only a single letter change and occurs quite frequently in the population, we call this a single nucleotide polymorphism, or abbreviated SNP. So what you see here on the slide is that person 1 at the position indicated with SNP carries a C and a T, while person 2 carries 2 Cs and person 3 carries 2 Ts. So this is an example of an instance where people may differ from one another in the genetic code that they carry. And such instances happen about every 1,300 nucleotides in our genome. Now you know this, you may wonder, so how do they get actually this genetic information? So we can get this by collecting saliva or cheek swab samples from children. And this is a relatively non-invasive manner, and these samples get sent to us often by mail, after which we are able to extract DNA from it in the lab. And this is actually a picture of the wet lab that we have here at the MPI. So once our researchers are able to extract DNA from the samples that get sent to us, we can infer the genetic code at each position in the DNA of an individual using very expensive machinery and so-called genotyping chips. So after we have done all that, we are able to study around 8 million SNPs across thousands of children. And we do this to learn more about their genome in relation to their language, their literacy, but also their ADHD symptoms. So I wanted to end this little section about genomics with telling you why would we bother to do genomic studies. So we know that genomic analysis can better detect and control for bias than non-genomic analysis. They also allow us to study trait relationships when these traits are not assessed within the same individuals, which allows us a bit more flexibility in the samples that we have to collect. And we can study it at a single variant level. So here we would investigate, is there a particular SNP that is associated with language development? But we can also study across the genome and then use it as a proxy for certain behavioral processes. I think you know now enough about genomics to follow the rest of the talk. So I will hand over back to Beate.
Thanks so much, Ellen. Yeah, that was a really nice introduction. But how can we now use genomics to better understand these links between language abilities and ADHD? So we do use a concept quite often that we call heritability. And that is the phenotypic variance of a trait, such as language or any other trait, or ADHD. And we characterize which proportion is due to genetic influences. In particular, heritability is defined as the ratio of variances. That means the proportion of the phenotypic variants that can be attributed to, here for simplicity, additive genetic influences. And we use a little sign for this. So here we have an assumption that carries all our phenotypic variation, which can be split into genetic and environmental influences. And this ratio, this H square, is what we call heritability. So now, what do we see regarding this heritability, especially for ADHD? So this is something that has been carried out by large multi-center studies. And it has been reported that the heritability for ADHD is modest but present, about 14%. So we know now in our little overview here that ADHD certainly has genetic contributions besides non-genetic contributions. So what do we know now about language and literacy? Also there, our own study, actually led and conducted here at the MPI, has also shown that mid-childhood traits and also in early adolescent traits are heritable. And this may include very different traits such as reading, this may include non-word reading, spelling, which we always call literacy together. So if we go back to our overview slides, we can also see that language and literacy is a heritable component beside non-heritable influences. Now, what about this link? Is this link between ADHD and language and literacy also to some extent due to genetic factors? And this is another study that we carried out here at the Max Planck Institute, where we studied the overlap between ADHD polygenic influences and language and literacy, in particular, the variation that can be explained by these polygenic influences, which is here described as regression R-squared, a multivariate analysis technique. And what you can see here, that there is definitely what we call an association, an overlap. So genetic influences that are associated with ADHD are also shared with language and literacy. So coming back to our little overview slide, we can see here that ADHD and language and literacy share genetic influences. Sorry, we had a delay in our slides. We want to know now, however, what the overlap is between early life vocabulary and ADHD. So what's the best way to understand this? Actually, we thought we may take a step back and look what we already know about the genetic influences that contribute to early life vocabulary. And we have carried out quite extensive research that looks into the overlap between early life vocabulary and later life language and literacy outcomes. Now, let's talk a little bit about language and early life language in particular. When we start our life here on Earth, then we start actually with absolutely zero words. Nobody knows anything in terms of language when they arrive in this world. Our receptive language starts first. The first words that are understood may arise already between the ages of 6 to 9 months. This is the time of the first understood word. And then we develop also skills as word segmentation, or that we can discriminate between different language sounds. And this language understanding is often called receptive language development. So our language understanding is very, very closely followed by our language production. And we call this often expressive language development. It starts with canonical babbling at the age of 3 to 6 months. Then our first spoken words arrive at about 9 to 12 months, in parallel with communicative gestures. And then we have rapid growth in our vocabulary with two-word combinations and ever-increasing vocabulary size with more and more complex structures that contribute to our language abilities. Because this is fairly complicated, we have put forward a few icons that can help you and give you some guidance during our talk. When we talk about receptive language development, we have a little infant here. This is characterizing language understanding. And later on, we have a little toddler, also characterizing language understanding. And for language production, we have a little infant here at the age of about 12 months, and which is followed by a toddler at the age of 24 months. So how would we assess actually these types of language development, in particular vocabulary? We often ask the parents. That's the simple answer. There are several questionnaires, especially the MacArthur Communicative Developmental Inventories that we use, but there are also other instruments. And we ask mothers to report on items such as, can the child say, pool, tree, dog? And when the parents answer yes, we have a measure that captures children's word production. If you ask the parents about items such as pool, tree, dog, but ask, can your child understand these words? We obtain a measure of children's word understanding. So now we can put this all together with our genetics. In particular, we carry out multivariate genetic analysis. We have here, for example, several measures that we can obtain in children. For instance, during the age of 38 months, which is close to the age of 3 years. And we can look at word production, and we can look at word understanding in relation to language related traits. But now we dissect the phenotypic variance into genomic and residual or non-genomic influences. Here are the genetic influences, and here are the residual or environmental influences. And to give you a better picture, Ellen will talk now for an example.
Yes, so here I actually wanted to show you one of the examples from work that we published in 2020, studying indeed measures of word production and word understanding at about the age of 3 years, in combination with a measure of verbal intelligence at about 8 years of age. And what you can see on the right side of the slide is actually the heritability of these measures. So differences in word production and understanding were for about 15% due to genetic influences, whereas this was a bit higher for verbal intelligence at age 8, with an estimate just over 15% actually. And now with this method, we can actually see to what extent represent these boxes the same genetic influences or different genetic influences. So we start with this first genetic factor called A1 that is explaining differences in word production at 38 months of age. And as you can see by the arrows going through both word production and verbal intelligence, this factor also explains differences in these other measures. And on the right side of the plot, you can actually see how much of the differences it explains. And for both, in this case, that is about 9%. On top of this first factor, there were additional genetic influences that were linked to word understanding at 3 years, here denoted by A2. And interestingly, these influences were also related to verbal intelligence at 8 years of age, but there they explained a much larger proportion of the differences, with 45%. And as you may already have guessed from the height of this bar plot, there was little evidence for genetic influences that were specific to verbal intelligence and not yet already related to word understanding and production much earlier on in life. We then applied a similar structure to the environmental part of this model, and there we can see that both measures assessed at 38 months to some extent share environmental influences, whereas these are more specific for the measure at 8 years of age. So together we concluded here that early genetic influences are important and actually get amplified during development. In a follow-up study, we again applied the same design, apart from including now even earlier measures of word production, with one measure being collected at 15 months of age and an additional measure at 24 months of age. And what this actually shows us is that word production at 15 and 24 months share genetic influences. However, word production at 15 months was not so much related to word production at 3 years of age, word understanding at 3 years of age, or verbal intelligence at 8 years of age. However, the first genetic influences that are playing a role in verbal intelligence at 8 years of age can already be traced back to word production at 2 years of age, which is or at an age before we can actually administer IQ tests. And on top of this genetic factor, we also again found evidence for this factor I explained earlier to you that was identified for word understanding, here denoted by I4, which again got amplified and explained a large proportion of the variation in verbal intelligence at 8 years of age. We did not only apply this design to verbal intelligence, but for a range of mid-childhood to early adolescence language and literacy traits. And here I'm highlighting another finding for reading accuracy and comprehension at the age of 7 years. And this is actually remarkably similar to what I showed you for verbal intelligence. So again, the first, the genetic influences that are important for differences in reading at 7 years of age emerged already for word production at 2 years of age, as well as for word understanding at 3 years of age.
Thanks, Ellen. These are really findings that are still like even today. So what can we actually then see? If you look at it a little overview, we can see that the factors contributing to early life vocabulary, as we investigated, are quite diverse. We have factors that relate to infant word productions, toddler word productions, and infant and toddler understanding. So these factors were all in a different way related to our language and literacy skills in later life. So we have really quite different relationships. So does this help us to investigate the questions how early life vocabulary relates to ADHD? We investigated exactly this question as part of our meta-genome-wide analysis of early language within the EE consortium. We focused on 3 different outcomes. The first one was really infant word productions, which was defined across the age of 15 to 18 months in about 8,000 children. This is not only one cohort, this is a multi-cohort project, and we have really many collaborators and colleagues and participants who donated their information that made this project work. A second phenotype that was also collected across many cohorts was toddler word production, which we defined between the ages of 24 to 38 months, about 16,000. And lastly, we also investigated toddler word understanding across the ages of 24 to 38 months, in around 6,000. And for all these children, we investigated variation in their language abilities in relation to variation in their DNA, especially across 8 months. The first thing that we found again was that there is SNP heritability of early language. About 80 to 24% of the variation in vocabulary size is really attributable to differences in genetic information, here the SNPs. And now we also wanted to investigate the genetic links across early language. For this, we use a measure that we call genetic correlations. And this measures the extent to which the same genetic influences contribute to variation into phenotype. So what you can see first is that we have really quite a bit of overlap. We have a genetic correlation of about 70 years, which relates word production, which is very early in life, the infant word production, to word production in toddlerhood. And we also have a correlation coefficient of 0.67 in later life. That means across toddlerhood. So word production in toddlerhood and word understanding in toddlerhood is really closely correlated. But what we also see here, there is very little evidence for overlap between infant word production and toddler word understanding. It seems that we really capture their very, very different phenotypes. So despite genetic overlap, there are also distinct genetic influences during early life vocabulary development. So what does it tell us when we study now the genetic overlap with other traits? Ellen will tell you now more.
Yeah. So thanks a lot, Beate, for introducing this very large collaborative effort, which has been a truly amazing experience. And I'm very happy to tell you now some of its findings where we studied genetic links of early language with later life outcomes. So here we again studied the same measures as Beate just introduced to you: word production in infants, word production in toddlers, and word understanding in toddlers. And first we looked at overlap with spelling, word reading, non-word reading, and phoneme awareness, or which we would together label as literacy. And here you can again see evidence for developmental differences. So we see especially evidence for genetic overlap of word production in both infancy and toddlerhood for spelling and word reading. And there are some signs of similar overlap for non-word reading and phoneme awareness, but the effects were a bit less strong there and did not pass or strengthen significant thresholds. We found little evidence for overlap with toddlerhood word understanding, which is a bit contradicting to what I told you before, but we believe that this may have to do with the nature of the data and that we may just have not enough data to have the power to find such effects in the study design. Then we also looked at overlap with measures of more general cognition, such as intelligence or educational attainment. And the latter is actually defined as the number of years of schooling in individuals that are above 30 years of age. And so here we again found evidence for the differences during language development. So we found little evidence for any genetic overlap with infant word production, whereas we found evidence for positive genetic overlap with both toddler word production and understanding. And this actually means that toddlers that are genetically inclined to produce and understand more words are also inclined to have a higher intelligence quotient score or more years of schooling later on in life. And finally, we looked at overlap with different types of childhood behavior or neurodevelopmental conditions, including childhood aggression symptoms, childhood internalizing behaviors, ADHD, and also autism. So for autism, I can be very brief. We found little evidence for any genetic overlap with early language. But for the other three measures that we studied, the pattern was again quite interesting and showed developmental differences. So the pattern was similar across childhood aggression, internalizing, and ADHD, but it was most profound for ADHD. And what we found here is that infants that are genetically inclined to produce more words are also the ones that have more symptoms of aggression and a higher likelihood of developing ADHD. When we look, however, at word understanding in toddlerhood, we found an opposite effect. Although this did not pass on significant extracted here. This trend is in line with what we saw observed, but with what we know, for example, for literacy traits in middle childhood, where we know that children diagnosed with ADHD experience more challenges with acquiring literacy skills, for example. And this would be in line with that. Children that are speaking, or that with children that are understanding fewer words in toddlerhood have a higher likelihood of ADHD. And then for toddlerhood word production, we found basically zero overlap, and that has to do with the fact that this measure shares some genes with both infant word production and toddler word understanding. And if you sum this positive and negative effect up, you will end up somewhere in the middle around zero.
Thanks so much, Ellen. Yeah, so we were really intrigued by this pattern, but there was a little sting. It was, as Ellen just said, not so very significant. So we really wanted to know, is it real? So we carried out some follow-up analysis of these genetic patterns and studied individual level genetic data, but also language and ADHD symptom information in the cohort that contributed to our meta-regression, but never had direct access to this information. And the first thing that we observed, and it made us confident that there was indeed a positive genetic correlation in ADHD symptoms at the age of 18 and 13 years with respect to word production in infants here at 15 months. And on top of it, we could also observe this flip in genetic association patterns, such that there was an inverse association between the likelihood of developing ADHD symptoms and word understanding at 38 months. And as before, in the larger meta-analysis, observed that in this transition phase, there was very little evidence for really defined patterns. So what does it mean? First, we can already say that the links between early life vocabulary and ADHD are true, and that they also vary for different genetic influences that we can observe during early life and that contribute to the architecture of early life vocabulary. So we think that there might be an underlying differences in processes that we observe, which is captured by the genetic factors and the associations that we can see. So we proposed a theoretical framework of 2 stages of learning, language learning. During the first stage, this would be defined as the stage of where children learn to speak, and they develop speech as a process that is often also related to motor abilities and executive function. And during this very early phase, speech emerges, single words emerge. During a later stage, which we called speaking to learn, which is above the age of 24 months, children have already developed some language fluency, and they are able to combine 2 or more words. So our genetic analysis suggests that during this early phase, there is little to no link between infant word production and general cognition. And infants with ADHD may encounter fewer challenges acquiring word production. So they produce actually more words. During the later stage, the stage of speaking to learn, our analysis suggested that more toddler word production and understanding is related to higher general cognition in later life. And toddlers with ADHD may encounter already during this very, very early phase more challenges acquiring word understanding. So when we put this together in a kind of summary that actually deals with the fact what these genetic links are between language and literacy and ADHD, seen across development, we could say that yes, ADHD ADHD and mid-childhood and early adolescent language and literacy have definitely genetic overlaps such that they share genetic influences in the way that a higher genetic ADHD predisposition is linked to encountering more challenges in acquiring languages and literacy-related abilities. And when we just go back in time, then we see that ADHD and toddlerhood understanding follows a very similar patterns. Yes, there are shared genetic influences where a higher genetic ADHD predisposition is associated with more challenges acquiring word understanding. But when we look very early in infancy, then this association is different, which may be related to a different process. Yes, ADHD and infant word production share genetic influences, but in infancy, a higher genetic ADHD predisposition is associated with fewer challenges acquiring word production. And this was very puzzling to us, so we looked what other cohorts and other research groups found. And there is a very, very similar finding that stems from a Norwegian birth cohort, where a higher genetic ADHD predisposition is also associated with a younger age at home. Our future research now gets really informed by what we found today, and we are really interested to take this further. We wanted to investigate the change in genetic links between language abilities and ADHD or early development in much more depth. We also have started out novel large-scale collaborative efforts within the Eagle Consortium to investigate what influences on development of vocabulary. And of course, we are also interested in general how language development happens in children with ADHD. At this point, I would like to thank everybody who participated in this research. We would like to say a big thank you to all cohorts, to all participants, to all collaborators and funders. And we thank you for your attention and we open to questions. Yes, please ask us simple questions very straight away, so we are happy to answer them.
And if you do come across questions later on, then you can also feel free to contact us either via our email addresses that are on the last slide or via X, former Twitter, or Blue Sky, dependent on what you find easiest.
These are our email addresses, which Alan just mentioned. So please feel free also to contact us via this way. And yes, we are looking forward to hearing what you think about it.
So Ruth is asking actually, I think, a very important question, which I can ask on her behalf, perhaps to Beate. So when participants get involved in research, is it possible to get access to your own genetic data?
So the question, Ruth, this really depends in which cohort you participate. What we did, we only collected the information that the cohorts provided to us in the way that consent was obtained. If, as a research participant, you have given the consent to a study that actually is allowed to give you this information, then this should be possible. However, in most cases, cohorts have carried out recruitments that such a feedback is not possible. This has to do that in population-based research, we do not provide established information on clinical information, simply because we do not have depth that would cover such a feedback for clinical phenotypes. And ADHD is related to medical research, and as such, many cohorts that are population-based have excluded such a feedback. There are, however, cohorts that provide in recent years these feedbacks, and if you want to contact them and give this information, so please feel free. I'm not sure at the moment what is happening in the UK, but there are definitely large-scale efforts from original the UK Biobank, where very different consent forms and consent schemes have been established to provide those information.
Does that answer your question enough? Or does anybody else have another question?
Yeah, please let us know if you want any clarification or if we can provide you with something. It might be also interesting for people to know that we have Twitter accounts and Blue Sky accounts also for the consortium. Well, I leave this. I will read this question from Ruth again. So, is there no barrier in terms of maintaining anonymity? Ellen, do you want to answer?
Yeah, so I think this is actually a very important aspect in research, that all data that we study have been anonymized. So there's, at least for us, no way that we could trace it back to the individuals. And this is in general in research. Once the data is collected, people that actually use the data, often these ties linking it to personal details get broken, so that there is really anonymity guaranteed. And I think that's also a very important aspect of when once you donate your data for science.
Yes, nonetheless, genetic data is personal data, and we maintain the highest standards of keeping safe. We follow GDPR rules, the data really at a very strict and stringent level of access, unless we have consent through the participants and the cohorts and very strict data transfer agreements and access agreements in place. No one else, only us, can access the data, and no one else, unless agreed, has further access to this information. What we usually share is the information at the summary statistics level. There you can find also information from our studies, and researchers from other fields can actually contact us to obtain the summary information. And also here at the Max Planck Institute for Psycholinguistics, there we have a repository which is called the Language Archive.
But this type of data cannot be traced back to actual individuals.
Yeah, summary statistics is really a summary of multiple participants. So there is no way of tracing it back to an individual.
So we got another question about the difference between clinical trials and the type of data collection that we work with. So I think it's important that we don't collect data ourselves, at least not now. We work with data that was already pre-collected, with the same principles, I think, apply. I have to think about the differences with actual clinical trials, because I think they are huge.
Yes, but clinical trials often do not contain the genetic information. Clinical trials have a different purpose. Often that is linked to drug use. And when genetic data is there, quite often this is the main outcome of that study. There are some clinical trials that also investigate genetic predispositions on actually usage, but this is not the type of research that we are carrying out here at the Planck Institute for Psycholinguistics. We have specifically a study of language and literacy traits in population-based cohorts, and through the genetic overlap, because we treat symptoms as a kind of a dimension, we can see how we can use information from the general population to explain and better understand differences in children and participants which have a chance of having ADHD or have even a clinical diagnosis of having ADHD, and can better understand how we can understand this, giving, of course, the assumption that we can extend this symptom spectrum between general population and clinical outcomes. Thanks. Also, I saw quick information. We saw this, that there was an information about clinical trials. So we will look up what we have. Yeah, seeing here, yes, it's information there. And you can also read about the standards that we have applied here about the general data protection regulations, GDPR rules that we have implemented here at the Max Planck Institute. And there is also information about that there. But here at the MPI, we do not carry out trials. We are carrying out association studies. Maybe it's also good to highlight again that we are looking at genetic links. We are not looking at causation. We can only say that there is link overlap, but we do not know in which way the direction of such a link happens.
Yeah, and I think usually in clinical trials, there's like an intervention or a drug or a treatment or a therapy. And we work more with data that is collected as is. So our participants don't have to do anything. They were just asked how many words does your child speak? And that was it. There was no difference between groups or anything.
Yeah. And maybe also saying it right is nice to have or carry out the studies. It is that there is still quite a lack of information about how children with with a higher chance of developing ADHD develop language. Quite often this information about language is not there. It has never been collected. And as such, we can develop these kind of proxy measures of language that are purely based on genetic information to better understand how these processes may work in ADHD.
Yeah, so indeed, to confirm also some questions from the audience again, we are working with data that was already collected from birth cohorts, for example, from the UK or from other countries where they just follow children, sometimes even longitudinally, and ask parents to continuously fill in questionnaires or do assessments of behavior, but not a clinical trial. So I think with that, we can round off.
Yes.
Thanks very much for everybody that was watching and for asking all kinds of interesting questions.
Yeah, it was an absolute pleasure. So we thank you also for inviting us. As we said, please feel free to contact us. And yeah, we hope to keep in touch, and we will definitely follow ADHD Europe. So it was an absolute pleasure to be here. Thank you very much, and goodbye.